Hello, my name is Ramiro and I'll be doing the pineapple and enzyme lab. My materials are canned pineapple, fresh pineapple. I have the gelatin there, and 3 plastic cups, each labeled with fresh, canned, and no pineapple, with inside the tub of ice. I'll begin by putting 2 pieces of fresh pineapple into the cup and canned pineapple into the cup. So I'll begin with the canned pineapple, and then the fresh pineapple. My hypothesis is if the fresh pineapple is put into the gelatin, then there will be a catabolistic reaction because the protein in the fresh pineapple will break the gelatin apart. If the canned pineapple is put into the gelatin, then there will be an anabolistic reaction because the bromelain in the pineapple will not be as strong as in the fresh pineapple. I'm now going to put the gelatin into each cup, labeled as fresh, canned, and no pineapple. I will be observing the the reaction every 2 minutes for a 10-minute span. Starting now. So it has been 2 minutes, and now I'm gonna be stirring the gelatin inside the cup to check on them. Yeah, the pineapple's all the way at the bottom, and this one remains the same. So for the data table, under 2 minutes, I will input that the fresh pineapple is floating, while the canned pineapple has already sunken. And the control, which would be the no pineapple, has no reaction because there is no pineapple. There's no enzyme within the solution. It has been 4 minutes, and the solutions remain the same, as the fresh pineapple still floats, and the canned pineapple is sunken. Okay, it has been 6 minutes, and the solution remains the same, as the fresh pineapple still floats, and canned is sunken, while the no pineapple has no reaction since there's no enzyme. It has been 8 minutes, and the solution remains the same. Fresh pineapple is still floating, and the canned pineapple has sunken. Both the solutions have gotten colder. When I poured the gelatin in, it was it had just been boiled, and throughout the 8 minute span, the temperature in the solutions have decreased.
Yeah.
It has been 10 minutes, and the solution remains the same as the previous checkups, where the pineapple still floats, the fresh pineapple still floats, and the canned pineapple is at the bottom of the gelatin. Both have decreased in temperature. Well, all three. All three have decreased in temperature. Describe the differences between cups. So in the fresh cup, I had the pineapple. Well, the fresh pineapple was floating, and in the canned cup, the canned label cup, the canned pineapple sunk. And in the no pineapple labeled cup, there was no pineapple to test. What was it like when you started and at the end? So when I first started the lab, the gelatin was hot since it had just finished boiling. And throughout the lab, it kept getting cold. It began getting colder and colder. And at the end of the lab, the gelatin was cold, and at the bottom of the cup there was some semi-solid gelatin forming. The pineapple enzyme bromelain breakdown or build proteins. The pineapple enzyme bromelain is a catabolistic enzyme, which means it breaks down. And after looking at the data, what is the best explanation for the results of the experiment? So, the fresh pineapple's proteins allowed it to float in the gelatin and not bind with the gelatin and sink like in the canned pineapple. How could you increase validity? A way to increase validity would be to have clear instructions on how to do the lab, and to have exact measurements of each substances, as an exact measurement of gelatin, or temperature in gelatin and temperature in the ice. How could you increase reliability? Way you can increase reliability is by holding the experiment for a longer period of time, as opposed from 10 minutes. And then, how do different types of pineapples affect gelatin? Well, the fresh protein in the fresh pineapple allowed it to float in the gelatin, while in the canned pineapple, there was no protein to allow it to float, so it sunk, since it had processed starch and sugars. I accept my hypothesis because the catabolistic reaction that occurred in the fresh pineapple solution prevented the pineapple from sinking, and the anabolistic reaction in the canned pineapple solution sunk the pineapple because the gelatin was able to absorb the pineapple into itself, since it had a weaker bromelain. Address any trends you see in the data. So a trend that I saw in the data is that the gelatin kept getting colder and colder as each 2 minutes progressed, and this occurred because the cups were surrounded or were inside a tub of ice, and the coldness from the ice transferred into the gelatin. And then, how is this applicable to a real life situation? I couldn't really find an actual analogy for this, but I guess if you're trying to make gelatin while including fruits in it, pineapple wouldn't really be a good substance, since fresh pineapple, since it will break down the gelatin starch and will take a longer time to actually form into a semi-solid. And yeah, that's it. Thank you.